infection due to COVID. Last year, this time all second wave was going on. And majority of us lost some of our close relatives or friends like that. Right? Similarly, plants also undergo such kind of stress. Okay? As a result of which, majority of the plants do recover from infection, but some of them do not. And one of the mechanisms we talk about here is RNA interference, which is one of the adaptive immune response in response to viral infection in plants. Okay, and that is operating from since the plant evolution. Questions come: how do you see the infection? How many of you have seen ever viral infection in your lifetime? Anybody? Plant infection? Infected plant? Virus? Which one? Citrus canker is a virus. Anybody? Any plant with a virus? Tobacco virus spread in woods. Have you seen in anybody from village here?
So I think I have seen a viral infection on maize plant, but I wasn't sure if it just a viral infection.
Okay, well, I'll show you a couple of photographs of them. So, the symptoms are one such. So take it to long back with the human civilization. Look at this diagram. Thin rough spike showing lot of poliomyelitis infection. Not polio, but pox infection, smallpox. Okay, so close to 2,200 years ago, the same thing happened. Viruses not evolved today; they are along with their host they evolved. Okay, and you are seeing yourself alpha, beta, delta, omicron, so many things coming out. Probably all Greek alphabet should be over, but viruses not evolving. Right? And that is the what the biology of living organisms to exist. They lead to their next generation. And because they do come across some of the errors during the replication, you see a lot of variations. Okay? So they cannot give rise to the same generations and generations after. So this is one of the examples of the smallpox growth there. If you go to the temple, usually those sculptures are inscribed on the walls or some stones which people see on the daily life. Look at this picture. The lake indicates for poliomyelitis. This is very old, close to 2,100 years ago. What does it say? The polio was prevalent in those days. Okay. So this is again the co-evolution is the only concept by which we talk about virus evolution or the origin takes place. Similarly, in plant case, we have long data, some of the history, but this is one of the mysteries solved by a group of scientists, which was initially reported by a Japanese police. She wrote that forest dust stops in the village morning by morning, but the grass I saw in the summertime has already turned yellow. And any of you seen this plant can see from in your village or in jail itself you will find now. This kind of symptoms are very common. So scientists have resolved this mystery and they published in a very good journal called Nature. Okay. So this is also depends on not whether it is a very high quality what. If you can solve a mystery, you can solve a mystery by a normal or very high thinking way. It is always possible to use it for every publication. This is tulip. Okay. Tulips are usually monochromatic, means either red or yellow or any other color. So in 17th century, people started in Holland. They started getting this kind of tulip bud, much more attractive than the normal one, right? So people started cultivating them. It is something like if you are owning a tulip bulb of Viceroy tulip bulb with this kind of phenotype, you have to ship out this many, though there used to be barter system. So it is something like you are having a limousine in your garage. Your neighbors will see, okay, this person must be having a lot of money, right? So it was a kind of wealthy indication that you own a Viceroy tulip bulb of such kind of symptom. So when the business goes and people started getting in advance, imagine this is the whole crop land. So I take money from all of you because if you can get 10 of them, you will be owning so many things for your life, right? So I get from them, but this is the virus infected plan. Virus says beyond the limit, the titer goes up. When the titer goes up, you suck up. Deep or deep, more in plant nutrition. Remember, they are obligate parasites. The intention is never to kill the host. Because obligate parasite will always try to block the program cell death in animals or any kind of hypersensitive response in plants. Okay? Professor Nandi will be teaching you plant immune response later on, but I am giving a very brief introduction of this part. This is a tobacco mosaic virus infected leaf, and these three scientists are primarily involved in identification of the virus and to show that there is something called virus poison, okay, which you just swallow up and which can pass to the filter. Now look at this green and the pale green highlights, okay. So that is the zone of defense. So we have plants RNA dependent RNA polymerase. So the role of RNA dependent RNA polymerase is to copy RNA to RNA. Okay. So one of such RDRPs are located near those green islands that allow the virus to come in. They generate lots of siRNAs. That's what we learn today. How do they generate? How do they transport it? So what I see is tobacco mosaic virus. This is the pattern. The leaf, the green areas are strong with the plant defense. The yellow already has surrender. Broadly, you will, if a viral particle will have a capsule protein core, and inside will be either RNA or DNA, any kind of nucleic acid, cannot be both. Okay? So can you tell me viruses are cellular or acellular? What? Okay, so obligate parasites, right? So similarly, chlamydia also obligate parasites. Chlamydia, one kind of infectious pathogen. Chlamydia also obligate parasite, but they have their own KGP unique mechanism. Viruses actually depend on their host for their energy, and the battleground is transmission. You know transmission, right? So the transmission part is that even pox virus. Pox can do everything by themselves. Transcription, replication. Some viruses have evolved to encode some proteins which can recruit the replication machinery for the replication. Okay? Pox can do everything except transmission. So transmission, that is the main battleground for which they will work on. And they have given a lot of classification with criteria. So look, if you look into virus particle, it can be either rod shaped like TLV, they can be geminate with two particles together, like poseidon. They can be spherical, they can be bullet, they can be flexural. So whenever you see a sample, if you look under electron microscope, you get an idea what is the structure we are talking about. Now, structure hardly say unless you know the replication. So the committee, which is known as International Committee on Vaccination Viruses, they name them based on the replication. You know about alpha coronavirus, gamma coronavirus, these are same. So based on the molecule, you cannot say it belongs to which kind of pathogen to belong. Okay. So, but what kind of virus actually need? Minimum amount of protein I'm talking about. They are parasites, right? Carrying a large amount number of genome will always have an ecological constant on them. Okay, evolutionary constant on them. What that happen? Viruses are smaller in size. Majority of the cases take to some proteins which are role having multitasking. Right? So, it's, for example, one pen you carry here today, one notebook, if you can take all the lecture schedule, then you are fine. Or else if you carry every day one notebook and new pen, which is not possible for you. Same case, what viruses need first is actually duplicate. So it's a polymerization helicase. Helicase because all the way nucleic acid in cell will become either a kind of secondary or tertiary structures which has to unwind so that the replication cell go for DNA or RNA virus. So this is important for helicase will unwind, polymerase will add those nucleosides. For protein is important for encapsulation. The fundamental difference between plant and animal viruses is that plant viruses will have an additional protein called movement protein. Can anyone tell me why? Have you heard about polio virus? No. Similar, polio virus belongs to a group called picornavirus. Picornavirus also infect plant. If you know the genome organization, exactly the sequence will be similar, orientation, 5' UTR, polymerase, and then only one of them is movement protein they are missing in animal virus. Can anyone please think about why plant virus is the movement protein or animal virus do not need? Anyone?
In animals, metabolism is very fast.
Just look into the movement. How do plant cells communicate? How do animal cells communicate? Plant cells communicate. Yeah, kind of.
Diffusion.
Plasmodesmator.
Yes, again, I will go into it.
Okay. So plasmodesmator connectivity is very narrow. It doesn't allow more than a size to pass through the next cell. Viruses are larger than them. What they do, they go near plasmodesmator open the cell. Okay, so that the whole virus can go from one place to other place. So this is one. And in case of animal virus, animal cells do not breed. Animal cells communication is right. Okay, so these are evolutionary. Now, if you understand that plants evolve on this universe due to animals, slowly viruses which have parasitized to the move towards the animal system, they due to the process of evolution, they found that we do not need to carry this protein. And why will be carrying when they do mathematics class? The mathematics what do? Only keep it in your hostel, come to the physics class. Same way, they have to stop. Or other way, class 1 children goes to class 2, will not carry class 1 books. Over. The same way. So evolutionary. Then one of the protein of today's interest is silencing suppressor. RNAi or RNA silencing is an adaptive immune response which is very, very prevalent and takes care of the infections in a higher scale. Okay? But I have shown you symptoms of tuberculosis virus, Punic mosaic virus. Similarly, there are several viruses. You all see symptoms of infection because viruses input some protein which can block that pathway. And it has to do to survive. Right? So evolution always favors the brave and the competent. So these are some of the photographs of some insect species, anti whitefly, okra, these all can transmit viruses, but specificity varies. Okay? Not all viruses can be transmitted by all types of pathogens. I'm not going to discuss in detail here today, but please remember the specificity comes from the food protein. So virion. Okay. So that is what determines the whole process of infection. Now, if you summarize the RNA virus multiplication cycle, first and foremost the entry. Viruses primarily enter through insect, I have shown you those, right? Because they cannot get inside the cell membrane by a process even bacteriophage do it. Remember T4 bacteriophage sitting over a cell and the student becomes squeezing tail-cell fiber, they contractile. Okay? Now, plant cell wall, anybody has an idea how thick it is, plant cell wall? Nanometer, meter, how many nanometers? This is close to 1 micron. Okay? Now TND size is 300 nanometer. So given by that concept, TND cannot ever sit over the plasma cell wall and put it special, right? So all the way, viruses enter to plant cells only through wounds, and these wounds are created by insects. But insect doesn't know that they are carrying the virus. This is a very good debate now is happening. That insect, those who are carrying plant viruses, going through a lot of neurological disorders. But it is in your food. Do you always or do you sometimes suffer from food poisoning? You went to a good Pizza Hut and took a grab of pizza but the base was rotten. It's not your fault. Same, right? And let's look at the insect perspective. They are getting attracted to some plants which emit some kind of volatile substances. It's very interestingly viral proteins can plant different process of the other day. Plant differences mostly through a process called jasmonic acid. Okay, they release jasmonic acid so that the insect don't come and feed. Plant viruses have learned to enforce some protein which block the jasmonic acid pathway. They rather regulate or alter some pathway where some secondary metabolites are produced which will attract the insect. Okay, so similarly in this case, first is entry. Uncoating is very important. Imagine somebody, bad person comes and starts firing with a 50-47. And if you allow that to happen, then tomorrow you suffer full score. Agree with all of us? So the entry is the door. So uncoating means the person is taking 47 by the time, what's the name? Sipsonka jumps and holds the guy tied up and they are free. So if you don't allow the person to take out the gun, you must say, verbally how much they can do, nothing. But once you catch Sipsonka, you all of us will jam and pounce him. But if he can take out the gun, then you take. Similarly, viruses, uncoating is the battleground. If the virus can uncoat it, okay, you see a lot of cases vaccination targets that pathway. You don't allow to uncoat. The moment you uncoat, you nucleic acid, one molecule in one cycle in 45 minutes can give rise to 1 million. So once you allow the virus to replicate, once you allow virus to transcribe in one cycle, the battle is lost. Okay? So the primary battleground will be the viral uncoating to block it. So therefore, lots of transgenic approaches have been done in plant where you can block the uncoating procedure. Okay, don't allow it to uncoat. The nucleic acid should be always encased, and nucleic acid is the infectious part. You know that good experiment. Anybody? Common molecular biology. Beautiful affirmation.
Very McLeod stuff.
Yeah, and Harsi says.
So
if you look into those perspectives, 1940s, 1950s, same way. Plant virus, another virus, bacteria, all came similar kind of information. So your research will be contemporary. If I do now the research of 1930, I cannot publish anything, right? Because the research has gone far ahead. So similarly, replication, then more translation, assembly, spray to the new host, everything has to happen. This is the common aspect for us. Now, this is the whole process. As I said, 10 to the 6 to 7 billion per cell happens if you allow 1 PMD billion to replicate in one cycle. So by then, it is all about the defense. For example, you must have seen those who are frontliners work as a policeman or hospitals, majority of them victim of corona in the second wave. Why? The amount of titer they got is far less than you and me sitting in a home in a lockdown. Right? So it is all between the good and the bad. Coming back to this point. Instructor hold up one guy, right? The next guy came. Okay? And then Kashish will go again, neutralize. You are safe. Right? Part came. Your name? VEDIO. So it goes on like this. By the way, 40 people came, you could manage. If 41 came, you are dead. It's all between the good and the bad. you all should good people listening to this so carefully. So the class is going very smoothly, right? If all of you start shouting and start cross talk, I do not talk. The same only. Here also, it depends on if the primary establishment of fossa is done in a cell, plant will survive. Because virus replication phase is very fast. You can see 1 to 1 million, then out of 1 million, let's say plant has depended, which can take care of only 10,000 molecules. Right? Then the rest can easily replicate it, and it goes in better than PCR mode. PCR, you can think about. So when you talk about replication, the whole concept of RNA comes from here. From a genomic RNA, by virtue of RNA dependent RNA polymerase of the viral encoded protein, they copy from genomic to antigenomic, just complementary sense. Okay? Now this is the catch. When there is a complementary sense RNA, they form a double-stranded form. Now whenever there is a double-stranded RNA in a cell, mostly exogenously, cells will sense a threat. So once cells will sense a threat, they produce lot of proteins which are called dicells. Okay? So those dicells will go chop them into small siRNA duplets. The siRNA goes and can target. Okay? So you can easily imagine this double-stranded RNA is inducing siRNA duplets. This siRNA duplets, one siRNA will go and find complementary here and cleave it, right? But remember that 1 million story. It depends on the stoichiometry. How much virus can replicate, and how much amount of post difference can be generated. Okay? Now RNA is one of the adaptive immune response. Okay, but there are some other like R gene mediated resistance also there, which Professor Nandi will be taking when he talks about the immune strategy. But I am only confining to this part. Okay, so please have to understand, for our knowledge, the double-stranded RNA is a part of viral replication RNA induces. So viruses induces RNAi during the infection process. Okay, and then they are suppressed also. Otherwise, we would have never taken this class. Right? Viruses would have been eliminated millions and billions of years ago. Now, if you look into cell to cell, as all of you say, the plasmodesmata is the primary, the only connectivity between the two cells. So, virus protein bind to the RNA, take it to the plasmodesmata, and that is by virtue of the opening of the channel. Okay, this is very important for us to understand. Remember, plasmodesmata is narrower than the nuclear core. There are lots of good paper came which are advent of all molecular biology techniques like crystallography, structure biology. The paper came, if you read about adenovirus also, how adenovirus uncoating takes place, very nicely they drop into the nuclear core complex, histone comes, then the uncoating takes place, the DNA goes. Why? It's more important for a parasite, a living organism needs to give rise to the next generation. Okay? So they ensure that is done by the mechanism where post-protein play a role. They are completely obligate parasites. Now, but the questions come, how do they survive out of the obligate parasite phase? Okay? So you can store them in lab freezer for long time. I'll give you an example of a DNA virus phase. Insect vector giving delivering the DNA, the virion. This is the next step is the uncoating phase. Okay? This is the step we should look into this part, how we can block this uncoating. Once uncoating takes place, the DNA gets into the nucleus. So inside the nucleus, remember, DNA synthesis never happens in genome, right? So all DNA viruses must have an obligatory nuclear phase, except pox virus. I said you, pox have everything, but they depends on the cellular factor for the translation. That's why pox became a separately obligate parasite. Okay? So it's an example comes in your examination anytime. Any example of DNA virus which does not go to the nucleus is pox. RNA virus, can you tell me any RNA virus which goes to the nucleus? The replication? Any virus, fine. Any RNA virus goes to louder. You can hear me? Everybody, back comes. So any RNA virus, you all know it's really severe. HIV, influenza. They go to nucleus. They have an oblique nucleus. Okay. So these are some of the factors for us to know. So this is the DNA virus. Cannot go for DNA synthesis. Needs to go to the nucleus. Then Roger cycle replication. You know 5x174 and M30. This is a typical Gardner genetics book you will find. The way 5x172 replicates. So this virus can replicate in the nucleus. Then in the nucleus there are histones present. Histones will bind to this viral genome and form mini chromosomes. Why mini? Size is small. Now remember, do you know about epigenetic modifications like histone, lysine modification? So now imagine this becomes a subscript of epigenetic silencing and activation. Okay? So any kind of mini chromosome form, it can undergo transcription. Transcript form, they the viral proteins form, they go to the nucleus and help the export of the virion to the next. Why? You see all lot of symptoms of infection. Okay? We will be taking some symptoms I will show it to you. We will be taking. Have you seen this disease in your village or in your right? Transmitted by insect. So different types of symptoms comes out. This, look at this plant, it doesn't have any flower or fruits. So old plant. The fact is, viral protein can not specifically bind to microRNA. MicroRNA are which regulate the plant developmental path. So if the viral protein can bind to the microRNA, what happens in this case? Plant development compromised. So, and imagine if it is site specific, then in that case the plants have a lot of abnormalities. You see here, the leaves are showing cup, right, which is governed by MIR 166, 165 pathway. Similarly, the microRNA forms here. I show you the example here, the mechanism. You see this plant for Okra or Hindi, different kind of symptoms you find. These all are expression of viral proteins and their serendipity interaction with the host protein. Okay? Some places viral protein to mimic the host protein. Okay? So again in pumpkin, so yellow mosaic symptom. Green means a lot of defense. Yellow means compromise. Recent publications have come up to show that chloroplast has become a Trojan horse for plant virus infection. Similarly, same virus infecting sunflower and okra, bhindi, different types of symptoms of the You have seen corona, different people have shown different types of response. Genotype is very important. Okay? Which has a better role for antiviral immune response. Now, what is in sugarcane? I have seen some maize. So, sugarcane mosaic, sugarcane yellow leaf fall. So, these are very distinct symptoms. Okay? So, you see also apple, apple apple. You know, the application came 10 to a million years back. That proved that human gut has as many as 300 plant viral RNA. Okay? So, because it comes in the form of the food use, salads, right? But do plant virus infect human? Why not? Anything more precise? Can animal virus infect plant? Just think corona infection. Can animal virus infect plant? Can corona infect plant? If not, why not? Why not? And my question is this one. What are you? What is the mechanism? Very precise.
receptors.
Very good. So mostly, right, I have to come to this point. Animal viruses enter the cells by virtue of their specific recognition of receptor present on the cell membrane. Plants are cell wall. Where they are getting access to the cell membrane? Nothing to it. Plant viruses enter only through the wounds, which are breaking the cell wall. They deliver the whole virion into the cytoplasm. And RNA virus replicate in the cytoplasm, DNA virus goes to the nucleus. Okay? This is a very simplified structure. Now, coming to the story that yes, if you want, you can give an animal cell line with some plant proteins which viruses make and virus can replicate. But they cannot come out. They cannot come out. So every step is individualized and well done. So dapper you will find sometimes this kind of solution, coffee, cactus, music. So names are given based on the type of symptoms they produce. This is a killer virus. Everywhere it is present throughout the country. Just to give you a glimpse, nothing much. So the main story comes from here. The gene silencing, where RNA makes RNA, no protein is formed. That is the whole concept of gene silencing. And it started long back when scientists started looking into the function of cells prostate using silencers, where no protein is formed, but the transcript is getting degraded. So it all started a long established phenomena where people, and mostly fire, it contributed significantly in the antisense transgenic by C. elegans on those days in non-English. Okay. Now, before I go, I will tell you this story. I said you about the uncoating inhibition, right? Roger Beachy first time developed a transgenic. Look at these two plants. This plant overexpression for protein of virus. Okay. This plant is non-transgenic. Now he inoculated these both the plants with the virus, and the non-transgenic plants very symptomatic. You see, everyone. Here it is not. Why? Because the plant was overexpressing protein code. So as soon as the RNA uncoating takes place, RNA is naked, the protein goes and encapsulated again. You understand what I am saying? So if somebody holds this guy, he is opening the hand and again you are finding. So he can take out AK-47 from his there. Very simple. Okay? Same policy, same strategy Roger Beachy used. So the plant is happy, bearing all transgenic concept in 1986. Okay, so and in science, whenever anything happens, people tend to follow the same thing. And the strategy worked with very viruses, but it has due to the growth of RNA silencing. How? Okay. Experiments started with Jorgensen with a market company. Petunia itself is a very nice purple color, so they wanted to more deep in the petunia colors. So they took charcoal synthesis and then make a transgenic charcoal. They found in the subsequent generation some of the flowers are variegated, like you see darker color as well as white, and sometimes they are completely white. So just surprised, see what is happening. Look into this case, they found that the transcript is formed, but there is a degradation. Okay, in this case. So because the charcoal synthesis endogenous is blocking the transient transform synthesis, the whole concept they are given as co-suppression. And the story they started with as a post-transcription and gene silencing. So similar situation also was found. Looked into C. elegans, different people. Finally, Nobel Prize went to Fire and Melo, who showed this is the RNA. The outside is RNA, which is important for induction of RNA interference. Okay, so and it was also further shown by an experiment that, for example, look at this slide. Sense and antisense RNA once they are given it sense against the gene, muscle twitching, phenotyping effect you can see, antisense, and double-stranded RNA. Whenever they are given double-stranded RNA, the twitching effect takes place, and this went to the next generation. So the question was very simple, that it can go to the next generation, the small RNA goes, and the mechanism was not known there. It was found in almost all combinations. They are looking to, I am not going into the details. All of them proven in C. background. Overall message from those studies came that neither the sense or antisense nor the dsRNA is the trigger. Now you look at the virus case. Two molecules, those RNA forms, and they inducing the RNA interference. Okay? And trigger molecules act in catalytic amount. So what they did, they constrained as less as only 2 molecules of dsRNA. So they found it is not stoichiometric trigger. Even if 2 molecules of dsRNA can induce lot of effect of RNA silencing. And RNA silencing systemic moved down to at least F1 DMS. It will take lot of effort, and they found almost in all organisms. Now, remember, Drosophila genetics by then was much more advanced. So, PTGS and co-suppression was fast in plant. The same thing happened for dwelling in fungi, where neurospora cluster, they tried to increase the orange pigmentation. Okay? After some time, interestingly, fungus breach out. That means RNA starts. Okay? So, that is what is important. Then, mostly C. elegans give rise to the concept of RNA interference by Mendelian fat. We also know RNA interference is important for total regulation of chromosome architecture, which is known as transcriptional gene silencing. Okay? I will try to touch upon that briefly in the later part of the discussion. And most importantly, they are ready to their siRNA and mRNA code. Okay. So, what is the basic understanding happening here? Double-stranded RNA is the same in the form of viral dsRNA, can be transposon or exogenously as given by Mendelian pair. It can also form by aberrant synthesis RNA copied by RDRP. Then the dicer comes, dicer makes the siRNA duplex. One of the end can go and bind as a this complex, other can amplify. Here the role of RNA polymerase happens in dicer. Now remember, dicer here and dicer here are the different type in plants. So these functions are non-redundant, and you can easily have a plant mutant and you can identify which kind of small siRNA they are being generated. So if you look into this graph, the dicers, the domains given here, is an RNA-3, basically double-stranded RNA binding protein. Okay, so this can bind to the end, and then this red ribbon is the ladder. And RNA-3 catalytic domains are present here, so they want to start and then they make it. Depending on the location of the ladder and the binding of the cross domain, the size of dicer molecules generated siRNA varies. Some of them may be 21, some of them 22, some of them 23, 24. So if you show a plant and tell us, okay, in my plant with the dicer mutant I am getting 24 nucleotide more, then you can say, okay, this is the transcription gene silencing phenomenon. Nowadays, because of RNA-seq analysis, we generate lot of information. So predominantly from those analysis, you can come to know which pathway is activated. Okay. So what is more important to know? Another protein name is AGO1. Okay. AGO1. So this AGO has a most important role in the cleavage. From different sources it will come. What is important for us to understand that siRNA DFK, RDRP, by application by RNA dependent RNA polymerase. Because remember, when they given only 2 molecules of dsRNA, still it can generate some amount of RNA interference. So that is there is a state which is amplifying this SRNA. Okay, so that is only possible by RNA dependent RNA polymerase which are present. Okay, in plant toxicity there are different types of present. mRNA pathway, it is different than SRNA. Now, SRNA can be both exogenous and endogenous. mRNA are all endogenous in nature. It starts with the RNA point-to-driven transcription. RNA point-to-driven transcription is the formation of primary mRNA. And this primary mRNA, they folded the process into BCL1 and hyaline into small nucleotide, and then they keep and pass through the nuclear protein. This sRNA, the mRNA, depends on their location. They can act as a translation storing, or they can keep the CDFS. Now, what I am saying, imagine a viral protein can block this pathway, or mRNA biogenesis will be blocked. Okay? So, yeah, this is important. If you try to converge both the pathways, mRNA and sRNA, one of the important is that this complex which is formulated with the help of AGO protein are the ones which is following in the middle, it binds to the complex, and then binding of AGO1 are reached with this pathway, either the pathway, with the generation of different segments of virus. Okay? Now, mRNA is important for regulating the plant architecture, plant development of the pathway, and everything. Right? So, if you remember, if any viral protein can block function of this, if any viral protein can block function of microRNA or sRNA, what do you find? The difference is compromised. Similarly, the plant which will look normal, the same plant looks normal like tobacco. If the microRNA path is altered, plant will look different. So basically, we call about symptoms, right? So what is symptom is the external manifestation of the disease, and that comes when there are a lot of interaction between the viral protein and the host protein. Now, mRNA target can be validated by different mechanisms. Yeah, this is important for us to understand. In a normal cellular condition, when there is a normal RNA present, because cap binding 5' and 3' polyamide protein, RNA dependent RNA polymerase cannot bind and cannot go for any amplification. Okay. Now RDRP can go for de novo silencing. RDRP can also go for prime amplification. So remember, in cells, not all the RNAs or mRNAs are perfectly ready for transcription completion. Some of them could be aberrant RNA. So whenever there is aberrant RNA, may not be having a poly binding protein bind to the polyA to the 3' nucleus. The RDR is ready to bind, and by de novo silencing can make a double-stranded RNA. So the moment there is a double-stranded RNA in the cell will sense that, and then they induce the dicell. Dicell will come and keep the generation of RNA silencing. Now remember, the generation of RNA silencing siRNA is from this only. This siRNA can go and bind to the target normal RNA, and now RDR can use this siRNA as a primer and can lead to the prime amplification, formation of double-stranded RNA. So formation of double-stranded RNA is very, very important. Again, dicell will come and keep the normal RNA or the RNA silencing also can take place. Is it ready? You understand what I am saying now? Here? Agree? You will quiz your name. Now please. Agree? Yes? Good. Please listen. If you are feeling sleepy, have a cup of coffee or roam around game and come back. Because sleep is very inducing. Okay. Okay. Now, this technique is used. This is what fundamentalized study. Agroinfiltration is a technique. It's a very easy technique. Commonly used. People infiltrate plant leaves, and you can see the filter. This is number one. It's a Benzamina plant. So if you overexpress JP, and if you look at the UV, you see green fluorescence. Do you see green fluorescence here? So now I silence GFP by agro. I'll see in the UV it turns red. Why? Answer and have the passport to see. Why? If you silence GFP, the plant under UV looking red in front. This. Anyone? Any guess?
Because since GFP is silenced by agro, green is red is completely.
Right. Have you ever seen a plant under UV normal plant? Leaf. UV. Nobody? So no problem. This is because of chlorophyll photosynthesis. See, when chlorophyll absorbs two reasons. Blue, also the red color. Okay? But this is the chlorophyll photosynthesis. Okay, so you did not take the first step. Okay, we have it now. Good. So now, very interestingly, what scientists have done, they have infiltrated this small leaf with the anti-GFP constant. So GFP transgenic present, I give an anti-GFP, so this RNA will be there in the cell, and then will double strand RNA, dicer will come and chop an RNA, and then finally the transgenic GFP will be silent. So this infiltrated leaf, slowly see, the siRNA moves and this becomes red, goes to red. Beautiful, you can really see them under normal UV hand then and take photograph. The question comes, how does this silencing is occurring here? Entire leaf is, so that means it is a non-cell autonomous silencing. Okay? Why? Because silencing generates to a cell number 1 of the infiltrated leaf here, and then it goes from here. Infiltration started here, silencing from here goes to the hospitalization, and probably you know the role of minor veins and major veins in loading and unloading. So same way it comes, getting loaded in the hospitalization, then it goes to the different leaves. Okay? So scientists started looking into this. They have rationally total RNA, run on the page, and then they found small nuclei of around 25 nuclei those days. They found this happens also in the other transgenic close-operation. The bottom line is that if you are going to publish a paper and show, okay, I am working on RNA interference, the reviewers or other people will ask you, what is the evidence that RNA interference is working? You have to isolate small RNA from those infected samples, for any sample for that matter, run on a gel and show whether they have a small polysaccharide or not. Right here. Similarly in the virus infected plant also. Okay? So this dynamics continue to happen. So post transcription gene silencing where small size siRNA ranges between 90 to 25 nucleotide does occur, and it cannot be seen where there is no PTGS. So always in your plant or cell will keep a control. So right now, remember, gene silencing induces the viruses, target against the viruses and suppress the virus. People have done small experiments, nothing, sometimes you should go by this notion that on big instruments, in science, only to pair of lines are important. And the type of hand, like people have shown that SRNA moves in plant, by doing a process called grafting. Have you heard about grafting? Can anyone please tell what's grafting?
स्टॉक जो स्टॉक को जब जोड़ते हैं मतलब एक ब्रांच मतलब एक दूसरे प्लांट की ब्रांच है और वो एक दूसरे की है तो उनको जब जोड़ेंगे एक स्कॉइन होगी मतलब जो स्टॉक होगा वो स्टेम का पार्ट होगा और स्पाइन होगा वो दूसरा दूसरे
Okay, so
प्लांट की
is right. So
ब्रांच
basically, you do one thing. You make a silence plant, stop, non-silent cyan. Tie them up. So when there is vascular connectivity, silence means siRNA are full. So siRNA through flowing, they'll go to the non-silent plant, and then you'll see finally those are also silent. And get good quality. But of course, those are 1990s, and this is 2022. So that 1990, that mind was far ahead, but now people will say no, this is the old. Okay? So what I'm saying that always do not go after the instrument, just use mind and then it works very well. So they have done so that okay, GFP transmitted mammalian cell line, GFP siRNA, and if you see GFP anti-siRNA, the silencing is very nice. So these are our experiments. Plant also come up. People identify dicer. So some of the experiment was done in plant system, some of them in the dicer came from again the yeast system, some from vascular system, which can play with. Now, you see in plant there are 4 different types of dicer. So far has come up. Okay? But remember, with the new genome sequencing coming up, you may end up in getting more number of dicers than you do. Because recently we know there is a come up of DCL5. In one type of plant, only one characteristic DCL5 has been found. So broadly, DCL1 is for microRNA binding. DCL2 for viral siRNA production. DCL3 for heterochromatin silencing. So whenever you do an RNA-seq data, you will find majority of 24 nucleotide small RNA are coming from the heterochromatin region. And heterochromatin region silencing is governed by this process called RNA-dependent DNA inhibition. DCL4. So all these activities are well, you will tell them. What sound came out? When you are tired and want to talk, I am comfortable with anything, whatever you like. Okay? So don't feel bad. I mean, you are from morning here, or waiting for the lab to join. All of you give a lab? Yes. Anyone who is from my lab? Okay. So anyway, so don't hesitate. Very practically done. And of course. Okay? Okay, I don't go for this kind of basic information. This is important for you. Quiz number 2. Last bench. 2. This is AGO 1 in anadosis. The orthogonals are given here. So if you abrogate AGO 1 has a role in risk formation and 3 days. AGO 1 is just lysyl endonuclease. Okay? So if you abrogate function of AGO 1, what do you see? If you abrogate the function, if you don't allow AGO 1 to function, what do you see? Next. So previous bench. Yeah, 2. Answer. AGO 1 has a role in RNAi. If you make a functional mutation of AGO 1, what do you see? 2. Then you lose the lunch. I'll start again from the zero slide.
Development protein research.
There will be no discount. Then further plant will be
silencing and the double stranded RNA will not be recognized and it will not be fully contained
it's an mRNA, right? So basically both mRNA balances should be stored and PTGA should be stored. So plant is small and abdominal. The same way, if you know the role of this laptop, just take up this form. Use the presentation. So people do reverse genetic approach to do this kind of study. Agro1, Agro4, DCL1, all of them they role, and then you can prioritize this directly. So you can always identify. Suppose you want to know role of a protein which is not known earlier. You know, when you have this genome sequencing comes up, only 1 to 2% genes their function is known. 99% not known. So how to quickly use it up? Because viruses can induce gene silencing in a short span of time, you can use a viral vector and study the expression. So these are you know the genes which are involved. So far, if you look into this space, the fight always continues, and PTGS is the mechanism by which host can defend itself from the viral infection. Viruses fight back by virtue of their ability. They import some protein which called a suppressor of RNA interference. Okay. So so far, literature-wise, all plant virus must include a suppressor. Because RNA interference is the primary antiviral. Okay. So they define this given here. Only to remember everything. Now, quiz number 3. Imagine that plant where we have silent GFP which shows red, right? Why? Why this will red? Right. What is the molecular mechanism inside? RNA, molecular. In this case, physiology part. What is the molecular mechanism inside? Nucleic acid, and it has lot of siRNA, GFP specific siRNA. Now, I infected the bee, same plant, with a protein which can block this pathway, and then see the plant under UV. Which kind of color I will see? Anybody? Is it GFP transgenic? Transgenic GFP, trans red anti-GFP, and you give a protein which can block this silencing.
Right.
Answer and get a passport to school. How many of you say red? How many of you say any other color? You hesitate.
Right.
Violet.
Right.
Red. You wearing Puma red t-shirt. That's why.
Yes.
No. Anybody? Question is very simple. Can anyone repeat the question? Let that tell me. What is the name? By the way. Huh?
AK Aman.
Aman. Aman, बताओ. क्वेश्चन क्या था?
सर एक प्लांट था जिसमें साइलेंसिंग GFP हो रहा है, तो उसमें वो कलर कौन सा एक्सप्रेस हो रहा है?
This is the plant. Okay. This is the benzamina, GFP, fluorescence under UV. The same plant when you silence with agro with GFP turns red because of chlorophyll autofluorescence. Now you give a protein which can block this silencing process. What you see here? Which color? Question is which color? Okay, but don't sleep. You are right. That's the way it comes. That's what we do in the lab. Suppose you want to know in coronavirus which protein is suppressor. Plant-based system can be done. You isolate all these protein, okay? Or you can clone them in a 35 vector and then put it to the plant. And then you will find in this case the green color comes back. This is called reversal of GFP silence. In the first case it was GFP was silence. So reverse the silencing process. Okay, that is called reversal of GFP silencing. And depending on the strength of the protein, you can say which is more strong suppressor, which is weak suppressor. Okay. Okay. So all these cases we find the genes which are the major role. Now look at this plant. The plant, the virus, when you give a higher amount, it is lethal almost. And scientists have identified which is the suppressor. Okay? So now they have done some mutation in the suppressor. So if you know my right hand only works, you chop off my palm, what happens? I control. Right? So they have taken the suppressor. What we find? Do you see symptoms here? Mild symptoms? Slowly the plant loses number. Because the silencing suppressor does not exist. This is other very simple. Viruses with mutations in the suppressor fail to build them. So remember, when a replication takes place, you will find n number of combinations of molecules. If a coronavirus particular nucleotide, that how many combinations of mutations can not you can see. But evolution favors only those which can survive in the cell. Okay? And also can go to the next cell or the next individual. That's the reason some of the Omicron variants are quite mild and Delta was very bad. So, so all these cases, now who is number 4? Susamda. These are all suppressor given, okay? You've seen the leaf diagram, the mutated virus. This is 35 years, where the silencing has taken place. Now you give a protein, all of them, and you see different degree of defluorescence. What does it indicate? Strength. Good. Okay. You. What is your name? Yeah.
Jyotsna, sir.
Josna. Josna. So, from these all 6 leaves, from where you can get maximum amount of siRNA, GFP specific siRNA.
First, sir.
First one. Everybody agree?
Yes.
And you, whatever, what is your name? Josna, you forget your name.
Vinay.
Vinay. Where you will see the minimum amount of siRNA?
Second one.
Huh?
Second one.
Why?
Fluorescent color get green.
Red light. Aman, red का नहीं बोला.
Sir, I think
ठीक.
fifth.
हाँ?
Fifth. So इसमें दोनों कलर एक्सप्रेस हो रहे हैं.
You start. You. What's your name?
Soumya.
Huh?
Soumya.
Louder.
Soumya.
Soumya. Soumya, tell me, which one will be having maximum amount of siRNA and why? It is have to make it interactive, so you don't have to remember anything, just go to brain and you. maximum number of SRNA, first one. Minimum amount of SRNA here. Okay, why? High depression. Okay, so basically remember here, rate is directly proportional to the silencing, and silencing is proportional to the amount of SRNA generation. Okay, green is directly proportional to the reversal of silencing. So basically, if you have 10 different plants, 10 different leaves, you can infiltrate, you can come to know what is strong suppressor or cold suppressor. This has been done several cases. This is a couple of years back data, but we know almost every plant virus action. This is what I am to tell you. If proteins of P19, they form a homodimer like this. P19, two molecules form a homodimer like this. So this site is fixed. In this site, I can take this pointer. In this site, if it fits, I can take this other pointer also. So this is for microRNA. This can be for siRNA. So if I hold both of them with this, then both the function is stopped. So P19 can cradle duplex of siRNA or mRNA of 19 to 21 nucleotide. So in the cell, whenever there is a 91 to 21 nucleotide, siRNA, mRNA nucleotide, P19 will go bind. So you see the symptom of infection because mRNA binding is blocked. You see the viral defense is compromised because siRNA is blocked. Okay. So these are all viral protein adaptations which has led to generation of such a lot of information. These are all done by all biochemical work. You can really do a very good piece of work to show that how this formation can be blocked. I am not going into the details, I will talk to the interesting stuff. Okay, quiz number 5. Not tired, no? Aman? I am the late guy, because you feel that.
No, sir, I wait for
Okay, so look at this publication date. Goes to 64 years. How more than that? Goes to 90 years. Right? So now, this scientist published this paper. They had tobacco plant inoculated the virus for tobacco rings for virus. Do you see this leaf showing spots?
Yes.
And do you see here in the spot? So he that time said, wrote in this article, usually and by an unknown reason, which is beyond my imagination. Initially the plants with symptoms, then they are become symptom free. This is the process for recovery. Okay. Recovery is a process because, I say, this leaf also contains siRNA. And always double-stranded RNA leads to generation of siRNA. So replication is happening concomitantly. Post depends also getting more. I was saying, 1% came, 2% came. So when the more number of bad people are coming, then we are compromised. If less number of bad people, we will never the bad people. Okay, now remember, when the bad people comes, more number, and couple of them we just to hypnotize, to become good. Or we tell them, ki, see, they are doing bad thing, at least you help us. And then they stand in our favor, and the host get activated. So this siRNA level, when it goes very high during replication, plants start recovering from infection first. So this recovery is because of the siRNA. And siRNA is homology driven phenomenon. Okay, now question to first bench. You, what's your name? Johnson. Okay, Johnson, this uppermost leaf are inoculate with tobacco rings for virus. Again, what do you see? What do you anticipate? And why? faster. Faster. What we anticipate is what? If I inoculate this leaf, everybody keep thinking. If you inoculate this leaf, re-inoculate with tobacco leaf for virus again. Imagine this plant generate sRNA, leaves are free of the disease. Now you inoculate tobacco leaf for virus again here. What will happen? You, what's your name? Divisha, tell me what will happen? You see the disease or not? What about you? You. So what is the name? Arthi. No. Komodani. What do you think? What will happen here? What do you think will happen here? Anybody? Volunteer. Faster. No, why?
So because the amount of SIRP is already high in those leaves, so as soon as the main inoculate with the virus, it will be clean and it will be infection.
So basically, what it is saying that because already there are a lot of SRNA against tobacco virus, if you inoculate this virus again, the plant will remain asymptomatic. Rather, it's a vaccination. Vaccine pollution, individual plant can be done. Now, question. I inoculate with another virus, let's say tobacco mosaic virus. Tobacco ringspot virus is different than tobacco mosaic virus. Now I inoculate this recovered leaf with tobacco mosaic virus. What will happen? Symptoms of only. Or why do you see so symptoms of tobacco mosaic virus? You are very right. I tell you some corrections. Why it will show tobacco mosaic virus symptoms? Very good. So RNA interference is homology region sequence specific degradation, right? So sRNA specific to only tobacco virus. Now, in this plant, virus is clear infection. Low. The people came down the heart for once them pieces like this. They are still mourning. They just get out of this place. Then their friend comes and opens the hand and they are visiting us. They put in that context. Yeah? So in this case, viruses are low, not completely cleared of infection. So now tobacco mosaic virus infection takes place. It replicates. It causes disease. So when it replicates, it has all the suppressors. Suppressors will also block the RNA interference which is already operating in the plant. So RNA interference goes down. Again, tobacco virus goes up. The plant will show dual symptoms of tobacco ring spot and tobacco mosaic virus. That is what we call synergism. You have heard this today, synergism. Very simple. This is called synergism. Okay. So this can happen. So this is okay. This is what I wanted to say you, that this is a phenomenon. The viruses do not always bring over the host comes there. You will find if you have a perennial plant in your garden, like biennial also like dolichos, which in Hindi called same or other. In one season they will be showing symptoms, next season they will not. They also are to some extent regulated by the temporal conditions, environmental conditions. A lot of role to play. Yeah, it is very, very true. So, super inoculation of the recovered tissue with the initially inoculated virus due to immune vaccination process, right? And resistance is sequence specific. So, if it is the other virus, resistance is broke down. Same happens with this jelly virus also. This mock means not put any virus. 10 days old plant, 21 days old plant, and 28 days old plant. That much better, right? So this is for recovery. So what happens in the DNA virus? RNA virus you understand. Can reduction in RNA form through replication. In this case, when the DNA virus replication takes place, the transcript can be overlapping, abandoned in the folded, clicked by dicer. Once they are clicked by dicers, the SRNA will form. One of the strand gets into the circumference, the other strand becomes the passenger strand, get degraded by cellular nucleosis like SG1. The amplification takes place. But what have you said? That generally by the transcript by form of overlapping, abandoned or folded, we can induce inside the same time. Now, if you suppose summarize what we have learned, it has a role as a mechanism, it can role as an antiviral defense, and most importantly function as immunity. Now please think about it. If tobacco reinforced virus can induce RNA interference in plant, and sRNA can generate across the length of the viral genome. So if you cut this place, add little bit of steel joint, don't imagine you're cutting my hand. I will protest always. Okay? You put the steel, it's become part of my hand. Right? So similarly, wherever it goes, it will be detected. So similarly, in viral genome will be viral vector. You clone, you put an MCS, you know MCS very well. You put an MCS, then you clone a gene of interest. How? Antisense implementation. Now, that gene of interest can also replicate along with the viral genome. So when viral genome generates sRNA across the length of the genome, it will generate against that part also, right? And that is antisense to the sense transcript which is present in the plant. Okay, so let's say this steel plate, sRNA goes into plant, target Y genome. What will happen? dsRNA. So then that gene is compromised. So you see the phenotype. That is the advantage of virus inducing gene silencing. You can really use a robust technique within 1 month. High throughput technique, you can say this out of these 20 genes, these genes are for this function. Okay, so these are the options of Veda genetics. I said you, the don't to do any transformation, you allow the plants to grow and then do it. So for example, of the 20 genes, one of them gene has a role in developmental pathway, only developmental phase. Then if you are generating a plant through tissue culture, it will never germinate. Right? But in this case, you are allowing the plant to grow to a certain height and then inducing it. Okay? So as I explained you, viral vector will be carrying a copy of the gene to be silenced in the cell. Okay. Now out of the classroom. How many of you have seen the latest Jurassic Park on the Dominion? Nobody? Nobody? So this is the latest movie you guys have seen? This is the latest movie you guys have seen? Right, red guy.
Marvel.
Huh? Marvel fan? What is that? It's a movie. It's a film. Huh? Science fiction. So Jurassic Park Dominion, see all the genetic engineering. Concept of locust, making, then again, like genome editing. Somebody got Nobel Prize genome editing, right? Sushma ji, बताइए. Nobel Prize who got in genome editing last year? I want to look at me anymore. Anybody? Yeah, you can put Susan. Very good. You know, this is my only humble suggestion to you. Be up to date. Do everything. And you must be able to understand a newspaper from page 1 to page 8. If not, otherwise, this dear friend, because the moment you click on news, the AI understands which kind of news you like, they keep calling you that news. You don't need. At least you should understand newspaper. So coming back to that, you should be abreast with latest information wherever you go next time. Okay? Anyway, so that is talking about the biological vector. Then also genome part. Watch that movie. You will like it. This is nice. So I have not seen one thing. This is science fiction.
Yes.
So